Welcome to this video. Now this video is focusing on some very encouraging, it has to be said, new research just published in the scientific journal Nature. Most prestigious journal, and it has been peer reviewed, so it's not like another of these pre-release papers that are often talked about. Now the background here is that when people are exposed to the COVID-19 virus, there's a very variable response. Now we know that people that are older are more likely to get severe disease. We know that people with comorbidities are more likely to get severe disease. And it also seems basically fairly well demonstrated now that the viral inoculum is an important factor. Now the viral inoculum is the number of viruses that someone is exposed to. So if someone is exposed to a large amount of viral particles, more likely they will go straight down into the lungs where they can cause pneumonia. So that's a factor. And also another factor in determining the severity of disease that someone suffers from is their individual genetics, because some people are going to be more susceptible to a particular viral or bacterial infection than others. So these things were fairly well known. But this paper sets out to ask another question and answers another question. And that is, how severe a disease someone gets when they're exposed to COVID-19, does that depend on viruses and bacteria they've been exposed to in the past? Interesting question, because there was an outbreak of a coronavirus in 2002-2003 that caused SARS, the severe acute respiratory syndrome. And that virus, it's a coronavirus, so is there some immunity that's conferred? If someone had that SARS-1 in 2002-2003, does that give them a degree of immunity to the modern coronavirus that we have now, the coronavirus 2 that's causing the COVID-19? So that's an interesting question. And to try and analyse that, this study recruited people that had SARS in 2002-2003, but also compared them against people that did not have SARS in 2002-2003. So they kind of had their own built-in control group here. And they also found from previous studies that people had had the SARS, the coronavirus 1 in 2002-2003, still had these T cells that generate the immunity 11 years later. That was already known, and this group actually discovered that there was immunity 17 years later. Now, to cut to the bottom line on this video, if you don't want to watch it, what it looks like is that the immunity from SARS coronavirus 1, 2002-2003, it looks like there is still immunity to that and to the COVID-19 virus after 17 years. 17 years. So although we know that the antibodies produced to the coronavirus that we're now suffering from, the COVID-19, even though we know those antibodies don't last long, just a matter of months, it looks like, if it's anything like the SARS coronavirus 1, that the immunity from the T cells may last for, hopefully, as far as we know so far, 17 years. So this is actually remarkably encouraging. Now, if you want to know about this, I'm going to go through it now. If not, I've given you the bottom line. It looks like the immunity is much better than we thought it was. So let's crack straight on with what we're talking about here. Now, what happens is we have lymphocytes in the blood. Lymphocytes are these special cells that are particularly good at fighting viral infections. And we have two sorts. We have B lymphocytes and we have T lymphocytes. So they're both lymphocytes, but there's B and T. Now what happens is the T lymphocytes, imagine that's a T lymphocyte there. They have a large nucleus like that. So that's the T lymphocyte. Now what happens is there's a particular type of T lymphocyte will send chemical messages called cytokines to B lymphocytes. These are the B lymphocytes. And it's the B lymphocytes that actually produce the antibodies. These synthesize the antibodies. So these produce the antibodies. And the antibodies, of course, are the immunoglobulins. The immune proteins that fight the infection. And there are these Y-shaped molecules that fight the infection. These are the antibodies, or the immunoglobulins, producing millions, I don't know, billions of those, I guess, are produced. So what we have is that the T cells stimulate the B cells, and the B cells produce the antibodies. So the T lymphocytes stimulate the B lymphocytes to produce the antibodies. That's what's happening. But this is all quite specific. So what we have here, this is a picture from the physiology book I wrote a few years ago. So what we have here is this is an antigenic organism, in this case the virus. And there are T cells that recognize this particular type of virus because they have these receptors. So a particular sort of lymphocyte, that's the lymphocyte there, will recognize the virus. And then what happens is, now this is the example of B lymphocytes, but the same thing happens with T. So we have this viral recognition here. In essence, these lymphocytes, these B lymphocytes in this case, are already there, and the T lymphocytes are already there. And the antigen essentially selects the one that it fits into. And when that happens, when the virus fits into the lymphocyte, that with that nice particular shaped receptor that fits onto the virus, then that lymphocyte will divide many times to produce many millions and billions probably of these B cells. And these B cells are called the plasma cells, and you get huge numbers of those produced, and they produce large amounts of antibodies. So that's what's happening. So, but of course you need the T. That happens with the B cells and the T cells, because you need the T cells to stimulate the B cells to produce the antibodies. So we get clonal expansion in B cells, and we get clonal expansion in T cells, but these are the specific cells that are good for fighting that particular virus. So what we have here is we have a particular type of cell there that's not the right shape. That's the lymphocyte, it's not the right shape. That one's not the right shape, but that one is, so it fits on. That one fits on, that fits onto the virus. And when this lymphocyte, because it's the right one for the virus, the presence of the virus stimulates this lymphocyte to divide. And it will divide into a population of, in this case it's B lymphocytes, that will produce millions of other copies of this. And these will go on to produce the antibodies, all these antibodies. But at the same time, this original cell that's been selected here will divide, and it will produce a smaller population of several thousand of these cells here. And these cells are long-lived cells. These are the memory cells. So these cells here that actually do the business and produce the antibodies in this case, these are relatively short-lived cells, but these ones live for years, and they can carry on dividing as well. And so we now know from this study that they can live for 17 years in the case of coronavirus type 1. So that's what's happening. So we get this process of clonal expansion. A clone, that's the clone there. A clone is two cells with the same genetics. So all of these cells here have the same genetics as this original one that was found to be able to fight the virus. And as well as that, as the immune response goes on, this receptor becomes more specific. So the lymphocytes that fight the infection become more specific. So that's kind of the background science. So this study here, SARS coronavirus special T cells, special T cell immunity in cases of COVID-19 and SARS. So COVID-19, they mean the current infection. SARS, they mean the 2002 infection. And uninfected controls, people that weren't infected. So if we have the T cells, if we have the memory T cells, these T cells know exactly how to stimulate the B cells to make the antibodies that are so precisely engineered to fight the particular virus. This is all very, very precise engineering that goes on inside these molecules. So memory T cells are induced by infection, as we've seen. So there's an infection and that will produce memory T cells. And memory T cells remember how to fight that infection and can stay active, several thousand of them in the blood, remaining active for many years, as we've seen, up to 17 years. And the presence of these memory T cells will influence the future susceptibility and clinical severity of a disease. So if this virus comes along again, because we have these memory T cells, they remember how to stimulate the B cells to produce the antibodies, exactly the right antibodies, and they'll do that straight away. And that means you get a rapid immunological response. So people may not even get symptoms. So they might not even be susceptible. But even if they do, they're likely to get a very mild illness because the body already knows how to make the specific antibodies to this particular illness. Now, they took patients who recovered from COVID-19. So this is the current pandemic. T cells from recovered patients recognise multiple regions of the severe acute respiratory syndrome coronavirus type 2. So that's not really surprising, really. These T cells that have been produced in the last few months as a result of this infection are able to recognise the virus, bind onto the virus, neutralise it and kill the virus. So that's good. But then they looked at patients who'd recovered from SARS. That's the SARS coronavirus type 1 in 2003. They looked at those as well. Now, they had 23 of those, and they still possessed long-lasting memory T cells reactive to severe acute respiratory syndrome virus. So these cells were long-lasting in these patients that had had SARS coronavirus 1, way back in 2002, 2003. So they still possess this T cell-based immunity 17 years after the 2003 outbreak. Because they still had the T cells, they still had these T cells, so these T cells could undergo clonal expansion very rapidly. You'd have a large population of these T cells that could then stimulate a large population of the B cells to produce huge amounts of highly specific antibodies really quite quickly. So that's all looking remarkably encouraging. Now, this is even more encouraging. They found robust cross-reactivity to SARS-CoV-2, and they found it to the NP, the structural nucleocapsid protein. Now, what this is in the SARS, in the coronavirus, as you know, there's some RNA in the middle, and then that's surrounded by a protein-based capsule. So that's surrounded by that protein-based capsid. So these antibodies, or these T cells that the antibodies stimulated the production of, they shared cross-reactivity between the SARS from 2003 and the coronavirus from 2019. And of course, we know that we have these proteins sticking out of it, these reactives, these infective spikes, and round about that we also have a layer of fatty layer as well. But the point is there was robust cross-reactivity. In other words, what this means, and this is the key point, immunity to SARS coronavirus 1 that caused the 2002-2003 outbreak was giving immunity to people that was giving immunity against the SARS coronavirus 2 that's causing the COVID-19 outbreak. So that's good. Immunity to one virus seems to give immunity to another virus. So that's encouraging. Not entirely surprising though, because they are very similar types of virus. So that's not too surprising. But what was surprising is they frequently detected SARS coronavirus 2-specific T cells in individuals with no history of severe acute respiratory syndrome, no history of COVID-19, and no history of contact with patients with SARS or coronavirus COVID-19. So this is really quite amazing. This means there's a group of people who have immunity to the coronavirus that caused SARS in 2002-2003 and to the coronavirus type 2 that's causing the current COVID-19 pandemic. They have immunity, they have the T cells that know how to recognise the presence of the coronavirus, the COVID-19 coronavirus. And these T cells know how to stimulate the B cells to make exactly the right antibodies to eradicate and neutralise the virus. Now the question is, where on earth did these people get their immunity from? But before we look at that, it could be a complete answer to this question. Why do some people get exposed and are completely asymptomatic? Why do other people get exposed and die? Well, this is probably part of it, a big part of it. It is the pre-existing immunity that they have to the coronavirus, even although they haven't been exposed to SARS coronavirus 1 or SARS coronavirus 2 causing the current COVID-19. Isn't this getting intriguing? So where did they get this immunity from? Right, in these people, T cells were active against different parts of the virus. So there's different proteins in the virus. So what they're saying is people that have recovered from COVID-19 recently, their T cells stimulate the production of antibodies that fight one particular type of the virus. But these people that are already seen to be immune, their T cells are stimulating antibodies that fight other parts of the virus, other proteins on the virus. Now, the obvious question, and we've looked at this quite a few times on these podcasts, is this related to the common cold? Because there's four coronaviruses cause the common cold. So is it how likely you've been exposed to these common cold viruses? And this was one hypothesis that was put forward to explain the way that children are often have a mild infection, whereas adults have a more severe infection. Could it be that the children have been exposed to the common cold causing type coronaviruses more recently? Well, this study seems to show not. Now, the epitopes are the bit on the antigen that the immune system recognises as being foreign. So the parts of the virus that the immune system recognises as being foreign. But they had very little similarity to the common cold. So the SARS coronavirus 2 and the coronavirus that causes the common cold actually are immunologically not very similar viruses. So the SARS coronavirus 2 that we have now, causing the COVID-19, is similar to the SARS coronavirus 1 that we had in 2002-2003, but it's not. The current COVID-19 virus is not that immunologically similar to the common cold viruses. So it appears that is not the answer. But what they did find in animals, there's another type of coronavirus called beta coronavirus. Now, beta coronavirus is a common virus that causes infection in animals, normally infecting the gastrointestinal tract of animals. An infection, if people have been infected with beta coronavirus that they could have incidentally picked up from the gastrointestinal tract of an animal, this induces multispecific and long-lasting T cell immunity to the structural protein, to this nucleoplasmid protein. So in other words, there's a virus that doesn't really make people sick, makes animals sick, doesn't really make people sick. But if people have been exposed to that, it looks like this has caused an immunological response in people, and that immunological response in people and animals is long-lived. It's a long-lived response. So could it be that part of the reason that some people get asymptomatic disease and part of the reason some people die is not just age, not just comorbidities, but could it be that the people that have been exposed to beta coronavirus from animals have already got immunity, this cross-immunity phenomena, to the current COVID-19 virus. Interesting. Also interesting possibilities for prevention. And this virus, this beta coronavirus, is common in cows, dogs, bats, and rodents. Now, personally, I avoid contact with rats, and I don't have a ferret. Contact with bats, very unlikely, although contact with bat feces is not that improbable. Personally, I don't work on a farm, so I don't come into contact with cows, but I do come into contact with dogs. Love dogs. Dogs are great. Could it be that people have been exposed to dog beta coronavirus and that's induced immunity? Now, this team conclude by saying, understand pre-existing T-cell immunity in the general population is of paramount importance for the management and of the current COVID-19 pandemic. So this is vital to understand. So what we're saying is there's a group of people who seem to have these T cells, these T lymphocytes that recognise coronavirus, that's got cross-reactivity with the coronavirus we currently have, even though they've never been exposed to it in the past. They've never been exposed to someone who's been exposed to it. They're not contacts, and they weren't exposed in any way to the 2002-2003 outbreak, but they do seem to have this level of immunity. So the obvious thing to do now, and I have written to Tim Spector's group about this, although it's quite hard to get through to him. If anyone sees Tim tonight, ask him. What we need to do is have a question on the COVID symptom tracker app. Have you been exposed in your line of work? Have you been exposed to bats, rodents, dogs or cows? So is it, for example, that dairy farmers and beef farmers are getting way less severe disease? Is it the case that dog owners are getting less severe disease? All interesting questions. But regardless of the answer to these questions, this immunological knowledge that we've learnt about the SARS coronavirus 1, to me, makes it likely, because the SARS coronavirus 1 and the SARS coronavirus 2 are so similar, makes it likely to me that even though the antibody response to SARS coronavirus 2 is very short-lived, the T-cell response will last, I would hope, for many, many years. Therefore, I think this adds significant weight to the argument that immunity to coronavirus 2 causing COVID-19, once someone's had it, is more likely now, as a result of this evidence, to be a long-lived immunity. And if immunity is long-lived, that makes me more optimistic that we will be able to eradicate this virus from human beings in exactly the same way that SARS coronavirus 1 simply no longer exists.